So the first item on our agenda, so to say, is the conversation about the DNA. Okay? So DNA consists of individual building blocks called nucleotides. So here you can see an entire nucleotide which consists of a phosphate group, a deoxyribose sugar, and the nitrogenous base. DNA contains 4 different types of nitrogenous base: cytosine, thymine, adenine, and guanine. Cytosine and thymine belong to the group of bases known as pyrimidines, and you must know that. The good news, you don't need to know the formulas. I'm not going to ask you to recognize what a particular formula, chemical formula, represents. Adenine and guanine, these guys, belong to purines. So you can see that in a nucleotide on the right, phosphate and the deoxyribose do not change. It's the same for every nucleotide that's in DNA. What is different between individual nucleotides are bases. Okay? And therefore, these bases is what they carry. They encode the genetic information in the DNA. What is the role of the phosphate and the sugar? They form what's called a backbone. When you look at a molecule of deoxyribose, it has 5 carbons, numbered from 1' to 5'. And once you start looking at the position of the phosphate group, you can see that the phosphate always connects the 3' carbon of one deoxyribose sugar with the 5' carbon of another. 3' carbon of one deoxyribose sugar with the 5' carbon of another. So we can legitimately claim that the DNA strand has a directionality. It means, think about, like, when you stay in line, say at the airport, normal people are not psychopaths. They face the reception counter, right? And the person behind them faces their back, and the person behind them faces their back. This is the way we stand, right? In a certain direction. DNA follows the same rule. Why is directionality important? Directionality allows you to read a text without any confusion. If you think about, like, if you are speaking English, then you read from left to right. If you are speaking Arabic, you read from right to left. But in each case, there is a direction. Okay? Now, DNA is a double-stranded molecule, as you can see here. Not just double-stranded molecule. It is a double-stranded helix in which individual strands are anti-parallel to each other. What does that mean? So if you look at this specific image right here, you can see that the left strand, if we assume that the direction is 5' to 3', right? The left strand its direction goes from top to bottom. And the direction of the right strand goes from bottom to top. This opposite directions of two strands is what makes DNA helix and type parallel. Okay? So, few super important things about the DNA. Well, probably the most important thing about DNA is that the bases that are the parts of individual nucleotides, they can form pairs. And they pair in the same fashion all the time. The A, adenine, pairs with T, and guanine, G, pairs with cytosine, C. So the pairs are AT, GC. So what forms the connection between individual bases? It's hydrogen bonds. The dashed lines in this picture represent hydrogen bonds. So I have a question here. The AT pair, the connection between A and T is weaker. So this connection is weaker than the connection between G and C. Anyone has an idea why? Yes. AT pair is held together by two hydrogen bonds, while GC pair is held by three. Now, it may sound as a part of DNA trivia, but it is not. As we'll see in the future, the proportion of GC pairs versus AT pairs can even give hints to scientists what is the role of a particular part of DNA. Okay? Now, an interesting twist on how we humans recognized that DNA is a double-stranded helix came basically from five-ish scientists. So you probably have heard about Watson and Crick that were awarded a Nobel Prize. There was a third dude with them, Maurice Wilkins, also got Nobel Prize. But their model, double-stranded model of DNA, was built on the discoveries of two other scientists. One of them was Erwin Chargaff. You don't need to know the names. Please do not memorize them, okay? So Erwin Chargaff was working with DNA. He didn't know the structure. And what he discovered is that the proportion of adenines, he did a chemical analysis, the share of adenines was always the same as the share of thymines, and the share of guanines was the same as the share of cytosines, which actually makes perfect sense if you think about that. Because where you have A, there's a T. And when you have T, there's an A. And the same is true for G and C. So I have a small intellectual exercise for you. So imagine you have a double-stranded DNA molecule, and somehow you have established that it contains 15% of guanines. What will be a proportion of cytosines in that molecule? So again, 15, yes. Can you also give me the proportion of adenines and thymines? Exactly. So it's 100% total. We already used 30%, right? So we have 70% left. We split it in halves. We have 35% of A's and 35% of T's. Does that make sense to everybody here? Like the mathematics of that. Now, I want to emphasize that this works only for a double-stranded molecule. Single-stranded molecule can have any proportions of A and C and G and C as it wants. Okay? So, since the information is essentially written in the bases, and the bases are hidden within the double strand, in order to read any information from that double strand, cell must be able to separate the strands. So the separation of strands that you see in the center is called DNA denaturation. Okay? So we, humans, also denature DNA for like research and analytical purposes. Let's say when we are running polymerase chain reaction. We use heat. Heat breaks down hydrogen bonds, strands separate. Interestingly, if we remove heat, these strands will come back, anneal together. Enzymes in the cell don't use heat, they use other mechanisms, but they also can split the cells. And since we are talking about the enzymes that work on DNA, enzymes must bind to a DNA molecule to either separate the strands or, read the information. So where do they bind? They bind in the grooves. So you see that the DNA molecule is not really, it is symmetrical, but doesn't have a central axial symmetry. So these big gashes here, called major grooves. The smaller gash is a minor groove. So, before we move on to RNA, I have a question for you. Do you think a DNA molecule is relatively stable or not? You don't think it's stable? I'm just trying to make sure. You don't think? No, double-stranded DNA, like from a human cells. Stable? You're right. We can, so to give you an idea, we analyzed Neanderthal DNA, which is 40, 50,000 years old. It's pretty old. DNA is stable. And if you think about the analogy that I gave you before, the cookbook analogy, it makes perfect sense. Because your recipe book has to be stable. It's not supposed to be destroyed easily, right? Now, RNA, in our cookbook example, it was the image on your phone, the image of the recipe. RNA, on the other hand, is not. And we will chat why that is important. So RNA is also composed of nucleotides, but it has some slight differences. One of the differences here is a different sugar, ribose. Now, if you compare ribose that is found in RNA to deoxyribose that's found in DNA, you can clearly notice one, and only one, difference. In DNA, at the 2' carbon, you have a lonely hydrogen atom. In RNA, it's OH. RNA molecule is unstable, specifically because of that group. And instability of RNA is great for a cell. Imagine that you made a copy of a gene, the RNA copy, but there were some mistakes made, right? So your copy of a recipe is not really authentic. Well, not a problem, because this copy of recipe will disassemble in a very short period of time, and you will replace it with the correct version. Okay? It's a pain in the butt when you work with RNA in the research lab, because you do all the work on ice, and yet RNA starts degrading while it's sitting on ice. Okay? Another difference between DNA and RNA is that in RNA, thymine is replaced with the with an nitrogenous base called uracil. So instead of ATGC pairs in RNA, it will be AUGC pairs. For our intents and purposes, RNA molecules are single-stranded. So there are no two individual strands, it's only one. So you see that single RNA strand. However, sometimes within that RNA molecule, there can be fragments, parts, that are complementary to each other, like this. So they will form an intramolecular base pairs. The structures are called hairpin loops. Now, it's often depicted this way. So you have two parts, like that's RNA molecule. Then you have the hairpin part, and then the loop part. So when you see some kind of a well-defined, well-developed structure, what F word should come to your mind? You say the word structure. Function. There's some kind of a structure, it has some kind of a function. These hairpin loop structures, practically all the time, have some kind of regulatory role. They may prevent RNA from being translated, or stop transcription, or allow transcription, and this and that. We're going to talk about this at the end of this unit when we talk about the regulation of the gene expression. Okay. Now there are 4 types of RNA. I want to emphasize this is not an exhaustive list. There are more different RNA types, but they kind of very niche. So messenger RNA, which carries the genetic information. That's exactly what we were talking about when we were, talking about the cookbook, right? The copy of the recipe. Make sense? That's your mRNA. Ribosomal RNA, sort of self-explanatory, part of the ribosome. Transport, or tRNA. So this RNA carries amino acids to the ribosome when the protein is synthesized. 2 types of RNA that students sometimes mix up are mRNA and tRNA, because they both carry. But while tRNA carries something tangible, i.e., an amino acid, mRNA carries information. Go ahead. Getting there. Just give me a second. Just give me a second. Yes. So, yes. Information. Carries amino acids. So think of it this way. Okay. In our cookbook analogy, let's pretend that we don't have cell phones. Okay? Good? So I'm kind of digging a hole for myself, but when you think about that cookbook analogy, you take a piece of paper and you copy the recipe on a piece of paper. That piece of paper, that paper carries the information about the recipe. Does that make sense to everybody? Now, when you go to the kitchen and get the components of your dish that you're going to cook, those components, let's say they are in the paper bags. So it's also paper, but this paper contains not the information, but it contains something tangible like flour, or salt, or sugar, okay? MicroRNA, I believe this question. For a short period of time, it exists as a double-stranded RNA. It's a very small bit of RNA that regulates the mRNA stability. I really like the analogy of it being a tag that you sometimes can see in the big office buildings. When they get rid of furniture, they put a tag, surplus. You know, like, we're going to throw these chairs away. So please take care of it. Does that make sense? So microRNA will literally bind to mRNA, and it will signal the proteins in the cell. This tag means destroy that mRNA away. Okay? Good? Now, we already mentioned the idea that the phenotype is determined by the genes expressed at the given moment. So in our conversations, when we use the term gene, a gene, we refer to a part of DNA that encodes, has a recipe for a single polypeptide, for simplicity, single protein, and that protein represents a trait that bacteria may or may not have. Okay, now let me give you an example. So, for instance, the ability of bacteria to use lactose as a nutrient depends on, I think, 4 genes. I can't quite remember the numbers, 3 or 4, doesn't matter. Okay, if either of them is absent, bacteria cannot use lactose. So if one of them is absent, the phenotype of bacteria changes. Absent or dysfunctional. You see what I'm trying to say? So the gene encodes a trait. Okay? Now, what about the relative sizes of genomes? Well, first of all, God forbid you from memorizing the numbers. Please don't. As long as you know this sequence of sizes. Viruses have the smallest genomes, then bacteria, then single-celled eukaryotes, then multi-celled eukaryotes. You're fine. Other exceptions from the rule, of course. Okay? Another important thing that you have to keep in mind, we are going to talk about bacteria changing their phenotype, altering their metabolic processes, for example, in response to certain stimuli. I want you to understand that it's all driven by gene expression. So the change in environmental conditions will result in the change of gene expression, which will result ultimately in the change of phenotype. Does that make sense? Okay. So now we're getting into the thick of it. So the idea today is to talk about the process of gene expression. And as a fun finish, I'm going to make a case for herpes viruses. Okay? So first, let's talk about how DNA exists in the cell. It exists in the form of chromosomes. If the chromosome is in bacteria, they are circular. And in bacteria, you usually have only one chromosome. Chromosomes in eukaryotes are linear. You can see linear eukaryotic chromosome on the left. And usually there are multiple. And not even usual, there are multiple chromosomes. The number can vary. It can be 4. Or it can be 72. I believe mice have 72 chromosomes. Okay? But the point is, it's more than one. Now, eukaryotic chromosomes, I'm going to point it up. Eukaryotic chromosomes can contain a fair amount of DNA that does not encode for any proteins. So some kind of a gibberish that is not a recipe. And we'll get back to the function of it soon. Okay? Now, it's important that to understand that DNA in bacteria and in eukaryotes doesn't exist as just the pure DNA. Let me explain. It exists being bound to a bunch of proteins. If we look at the DNA, chromosomal DNA in eukaryotes, it's bound to histones. And if we look at the chromosomal DNA in bacteria, it is bound to proteins that are very creatively named histone-like proteins. I mentioned it before. Folks, I understand, like I love this stuff, but I understand that for many of you, it may sound dry and boring. So hear me out. Here's a little clinical connection, not microbiological, but still. So first of all, why the fact that DNA is bound to proteins, why is this fact is important? Think of these proteins as bookshelves. Okay, when your book is on the shelf, when the DNA is bound to such protein, you can't read it. Makes sense, right? So you need to take the book from the shelf to read it, to open it up. In other words, you need to separate the DNA from histones or histone-like proteins so it can be transcribed, the genes can be expressed. Well, guess what? In many human cancers, the genes that are not supposed to be expressed, the genes that are supposed to be sitting on those histones, they are separated. And a lot of modern effort into cancer therapy focuses on the inhibition of regulatory proteins that separate the DNA and the histones. Make sense? So this is the fundamental knowledge. But if you can control that process, if you can regulate it as you wish, you can regulate cellular processes and potentially, reduce the cancer progression. Okay? Now, both eukaryotic and prokaryotic cells can contain extra chromosomal DNA that is not a part of the genome. In eukaryotes, it's a DNA in mitochondria and chloroplasts. We talked about it before. In prokaryotes, the DNA is found in plasmids. If you look at the image here, you can see that in bacillus, we talked about bacillus anthracis, plasmids, relatively small, circular, double-stranded DNA molecules encode toxins. And these toxins make bacillus anthracis pathogenic, dangerous. And if you wonder, what if we remove those plasmids? If you remove those plasmids, it's not pathogenic anymore. Okay? All good? The logic, does it make sense? So, very critical bit of information. Extrachromosomal DNA is not essential for a cell. So it adds some benefits, definitely in prokaryotes, definitely in bacteria, but let's put it this way. If you take these plasmids out of bacillus anthracis, do you think the cell can still reproduce? Oh, it can. It's not going to cause the disease. It may lose some evolutionary advantage, but the fundamental process of cell division will not be affected. Go ahead.
Can they be like hurtful themselves too?
Oh, then it will lose evolutionary advantage and will be pushed out. Like evolution will, result in its disappearance. But what's interesting, the paper was published a couple of years ago. Turns out, so scientists asked themselves a question: what about mitochondrial DNA? Can you take it out? Well, if we take it out, can a cell survive? The answer is yes. You can remove, I don't know how they did it, don't ask me. They removed not mitochondria. It's probably some complicated stuff, but they removed DNA from mitochondria. And cell was able to reproduce anyway. So it's not essential. Extrachromosomal DNA is not essential. Okay? And the genetic material effectively is replicated to get passed to the next generation of cells. Now, the first step of gene expression is to make that copy of the recipe, to make an RNA molecule using DNA as a template. We call this process transcription. If you think about this, the name makes sense. Are you familiar with the term like a transcript of a lecture? Somebody told, like, for instance, I'm telling you stuff in English, and then you convert the recording of my voice into the text, still in English, but a different medium. It's the text on the paper, not the text spoken word, right? So same here. The language is the same, it's nucleotides. But there are slight differences, such as different sugar, U instead of T, and so on. So what essentially happens during transcription? This. The enzyme called RNA polymerase synthesizes the RNA molecule using DNA as a template. And I'm going to, for now, skip the text and focus on this image. Okay, so this is bacterial RNA polymerase. In eukaryotes, there are 3 of them. I want you to understand the basic fundamental concept, okay? We're not going to go into pol 1, pol 2, pol 3 in eukaryotes, just RNA polymerase in bacteria. So it binds to the region of DNA, the region before the gene, that's called a promoter. You're going to be bored out of your mind with my recipe book analogy. But let's say you are going through a recipe book, like, flipping pages, and you get on a page. How do you know what recipe does that page contain without reading the recipe? Let's say you don't have content, huh? Like you have a page. How do you know that it's peach cobbler? It says on top, peach cobbler, right? That's a title. Title does not tell you how to cook it. It just tells you the text below is about peach cobbler. Make sense? Promoter is quote-unquote a gene title. So RNA polymerase binds to that title, to the promoter. And first it separates the strands. So up to this point, it's initiation. And then RNA polymerase starts chugging along, producing the transcript, producing RNA. It moves on until it hits terminal repeats, the termination sequence, and RNA polymerase stalls, falls off the DNA and RNA molecule is released into the cytoplasm. So far, any questions? So let's take a closer look. What's going on within the polymerase? So the blue lines represent DNA, right? Now you can see that there's only one strand called the template strand, which is transcribed. I'm going to pause and I'm going to look around and I'm going to ask you, does that make sense? So this enzyme, RNA polymerase, by the way, it moves in that direction. RNA polymerase reads only one DNA strand, only the template strand. Does it make sense? Okay. Now, what is the reason I told you that how do I know that it moves from left to right? Because DNA molecule is read from 3' to 5'. It's the law. Okay? Good? So it reads template strand from 3' to 5'. And since molecules of nucleic acids must be anti-parallel, look, it moves from 3' to 5', like reading 3' to 5'. But RNA is being synthesized from 5 to 3. Let me repeat it. Strands must be anti-parallel. So if you look at the direction of the template strand, this is 3', this is 5'. Do you agree with my placement of an arrow? Now, if you look at the synthesized RNA, 3' end is here, and 5' end is here. So it's all good. Freshly synthesized RNA strand is anti-parallel. But the synthesis, the new nucleotides, are added right here, at this end. So the synthesis goes from 5' to 3'. Okay? If you want to ask me why, go ahead. If I would know the answer, I would not be in this classroom. I would enjoy my private island somewhere in Bahamas. Also, you may notice that there are 2 DNA strands, but only one is read and one RNA strand is being synthesized. So if you will ask me, can we get rid, can we just hypothetically take template strand only and transcribe it? The answer is no, we can't. Transcription requires 2 strands. Do you understand what I'm saying? So 2 strands must be present. Only one will be transcribed though. Again, if you will ask me why, my response will be the same. If I would know the answer, I would not be here, okay? The easiest way for me to demonstrate it, there are viruses, which genome is actually one strand of DNA. When they infect a cell, the first step that they have to go through is to make second strand of DNA. Synthesize it. Once it's double-stranded DNA, it can be transcribed. That's it. It's the law. Okay, we good so far? So this is the story that this is how you should describe transcription, is a story. Okay? So in eukaryotes and in some viruses, the DNA molecule encodes, in addition to the meaningful part of it called exons. It also contains the kind of meaningless parts called introns. So this colored rectangles are exons, okay? And they're separated by introns. So if you make initial RNA, think of it this way. You have a recipe of, well, let's stick with the peach cobbler. But that recipe is sort of peppered by the fragments of the instruction how to cook crystal meth. If you will just go through, God knows what you're going to get in the end. So you need to somehow remove the crystal meth parts. Okay? So when cell transcribes DNA, it produces that primary transcript, the RNA that is not suitable for future protein synthesis. So this RNA goes through the process known as splicing, in which introns are excised, removed, and the exons are stitched together. Does it make sense? So again, splicing happens in eukaryotes, in all eukaryotes, but interestingly, it was discovered in the viruses, in adenovirus. Okay? So, splicing precludes protein synthesis, as you see. Transcription happens first, then there's splicing, then, only then, you can get protein synthesis. The advantage of splicing is that cell may not include all 5 exons, such as in this example, into the final mRNA. So check this out. This RNA contains all 5. These two each contain 4. Different 4, though. This one misses number 3, and this one misses number 4. This is called alternative splicing. So the proteins that will be produced, if you will look at these 3 proteins, A, B, and C, they have a lot in common, right? You see that? They have a lot in common. There are some parts that are missing, like this protein B, for example, is missing that part, okay? And the protein C is missing kind of that part. So that may alter protein function as a result. It may be located in a different place in the cell. It may or may not, it may have some other enzymatic properties or signaling properties or whatever, okay? Now let's say we are done with the splicing. By the way, before we move on, I have a question for you. A bunch of questions, actually. They're really straightforward, seriously straightforward. Do bacteria have nucleus? No. So when the DNA is transcribed and RNA is made, RNA, like where does transcription happen in bacteria? Cytoplasm. Bacteria don't have nucleus. So it effectively is in the cytoplasm, right?
Yes.
I mean, it's in the nucleoid area, but there is no boundary between the DNA and the rest of the cell. What about eukaryotes? Where's DNA? Where's the transcription then? Also in the nucleus. Where is splicing then? Also in the nucleus. So now think about, we're getting there. Think about the difference, fundamental difference between prokaryotes and eukaryotes. In prokaryotes, when RNA is synthesized, it's immediately available for translation, for protein synthesis. In eukaryotes, you have to make RNA in the nucleus, splice it in the nucleus, then get it out of the nucleus into the cytoplasm where ribosomes are, and only after that protein synthesis can happen. Which brings me to these important images, one of which has a gross mistake. Okay? So, let's take a look at this image first. The one on the left, okay? So this egg-shaped structure is RNA polymerase. This faint blue lines is DNA. Right now we are talking about bacteria. So DNA here in this example is being transcribed and mRNA is produced. You just told me that this mRNA sticks out in the cytoplasm right away. I'm not mistaken, right? So ribosomes are in the cytoplasm. These are ribosomes. They can jump on the RNA molecule while it's still incomplete, like it's being actively synthesized, but ribosomes already start translating it, forming proteins. So if you look at this part of the image, you can see all those RNA polymerases turning out purple mRNAs. And each mRNA is populated by multiple ribosomes. We call it a polyribosome. And each ribosome turns out a protein that this mRNA encodes now. Does that make sense to you? Do you understand what I'm talking about? And if you compare it to the process of protein synthesis in eukaryotes, you have to completely transcribe a gene, remove introns during splicing, transport that final version of RNA transcript, that mRNA, into cytoplasm, where ribosomes finally can read it. I usually compare it to the classic book publishing and people who write novels online. If you are a writer who publishes a book, you write the whole thing, right? Like you have to finish the book. When you finish the book, it's published as a book. You're not just selling chapter after chapter. Compare it to publishing, well, not even online, in some kind of a magazine. So you publish first chapter in January. While people read it, you write the second chapter and publish it in February. While people read the second, you write the third. So effectively, people are reading your story while you are continuing to create it. Am I making sense? So that concurrent transcription and translation, almost simultaneous transcription and translation, is unique to prokaryotes. The transcription and translation that are separated in time and space are a eukaryotic feature. Okay? So, now I often go a little bit bananas on the information. You will see when we get to sorry, I'm blanking. When we get to herpes viruses, sometimes just go bananas on information. I will tell you exactly what you need and not need to know. So please don't memorize the specific composition of subunits. At this point, all I want you to understand, any ribosome has a large and a small subunit, okay? And I want you to acknowledge that bacterial ribosomes are different from eukaryotic. That's it. It's a day different, okay? Now, how exactly is information coded in the DNA? Or we're talking about RNA, so we're going to focus on the RNA codons. Go ahead. It's structural. It's a very good question. So, okay, fantastic question. So, I'm going to ask you a question that is, it's not complicated. If you take a human gene and we'll try to, I don't want to say the word feed it, but let's say feed it to a bacterial ribosome, will it translate a human gene? It will. Ribosomes don't care what specific sequence to translate. Bacterial ribosomes need other different signals to start translation compared to eukaryotic. But functionally, all they do is making protein. Structurally, they're different. And when we get to antibiotics, this information becomes very important, understanding the specificity of like half of all antibiotics that we use. Okay? That bacterial ribosomes are different from human, for example. But let's go back to our encoding issue. So human proteins consist of, now, honestly, I'm being a show-off here. If you took biology and remember that stuff from there, every textbook tells you that there are 20 different amino acids. There's 21. There's an amino acid called selenocysteine. It's rare, but it's present in every living organism. Okay? So, whatever. 20, 21, it doesn't make a lot of difference. So effectively, think of it this way. You need to name 21 objects. You have a language that contains only 4 letters, A, U, G, and C, or A, T, G, and C. Do you follow my logic here, okay? So there is a minimal length of a word that you can have to encode. If you try to encode each amino acid with 1 letter, it's not going to work. You have only 4. If you're going to try to do it with 2 letters, the possible number of combinations is 16. Again, not enough. So the code, the genetic code, is comprised of 3-letter codons. So these are different codons. Each codon always consists of 3 nucleotides. Okay, now I'm not going to move on to the next statement that I'm about to make before you all confirming to me or like agreeing that you have no questions, like it's understood. So each codon consists of 3 nucleotides. Does that make sense? If you take any codon, except for the stops, but we'll get there. Take any codon. It always encodes 1 amino acid. So codon UCC means it's serine amino acid. Not histidine, not proline, not leucine, serine. So if you take any codon, you can assign a single amino acid to it. Make sense? However, a single amino acid can be encoded by more than 1 codon. Threonine is encoded by 4. Alanine is encoded by 4. The most outrageous is leucine, which is encoded by 6 different codons. Now, this is very similar to human language. If I tell you that something is large, do you clearly understand the quality that I'm talking about? Right? If I tell you an elephant is large, there's a clear-cut statement. But I can tell you that elephant is huge. It's humongous. It means the same, just different words. So the same idea is here. That existence of, let's call them like synonyms. We say that the genetic code is degenerate or redundant. So each amino acid may be encoded by 1 codon. Okay? They're methionine and tryptophan are 1, but whatever. Also, there are some stop codons. Okay? Here. So for the exam, please memorize the entire genetic code table. I'm just kidding. Don't even think about it, you know. Like, I quite literally remember a single 1 codon. It's CCC. It's proline. That's it. If I need to know a codon for any other amino acid, what I do, I Google. Nothing wrong with that. I'm not going to ask this, okay? Does that make sense? So, what is the mechanism of recognition? How does a ribosome, quote unquote, knows what to put in? So this is mRNA. And this is the codon GCC within the mRNA. Do follow so far. Let's take a look at the table and see what amino acid GCC encodes. That's your GCC. So it's alanine amino acid. We talked about 20 minutes ago about tRNA, transport. The RNA molecule that has this poetical clover leaf structure, okay, that carries amino acids. So in this specific case, an amino acid, alanine, would be attached right here. Now, if you look at this loop, you can see that at the very tip of that loop, there's a sequence C, G, G. Can you all see that? That sequence is complementary to GCC. So this is codon, and this is called anticodon. Okay? Nice. Right? So, before, I want to ask you, do you understand the process of quote-unquote selection of how amino acids are selected? I want you to understand something real important. I know that one of my many weaknesses is the fact that I make cellular processes very anthropomorphic. It's like, they want to do this. Nobody wants to do anything. It's just chemistry, okay? So here's the idea. If anticodon and the codon match, amino acid is inserted. If they don't match, the tRNA will be kicked out of ribosome until the proper tRNA comes in. It's a random process. It's like, imagine that you have 20 keys and you need to open a specific lock at a given time. And you're just randomly poking with the keys and one of them is going to fit. If it fits, you open the lock and you move on to the next lock and find the matching key again. Does that make sense? So we are now at the point when, you have your proper mRNA, whether you bacteria or eukaryote, doesn't matter. So you need to signal to ribosome that ribosome should jump on that RNA. If the ribosome, we're talking about prokaryotes, they mRNA have so-called Shine-Dalgarno sequence. Shine-Dalgarno sequence is a signal for a ribosome, hey fellas, jump on, start translating. Eukaryotic mRNAs have a so-called 5' cap structure, which has the same role. Ribosome recognizes 5' cap structure and starts translation. First, it has to assemble the translation complex. That translation complex consists of, first, small ribosomal subunit, which binds to mRNA and the first tRNA. Once that binding is complete, and tRNA, the first tRNA, binds to a start codon shows where translation should start, which is AUG in both prokaryotes and eukaryotes. Okay? One common confusion that students have, they start mixing up things like promoter and start codon. Promoter is the start of transcription. Start codon is the start of translation. We're good? All right? So once they're all assembled, large subunit joins the party, and you have this. This is the initiation complex assembled. So what you're looking at here is one step somewhere in between the initiation and termination. So you see this tRNA that enters that pocket. The pocket is called an A site. This A for attachment. The site in the center is called the P site for polypeptide. You can see that there's a tRNA with the polypeptide chain hanging off of it. So when the next tRNA comes in, the entire polypeptide is transferred onto the incoming amino acid. This little dot is an amino acid. And the ribosome shifts. When the ribosome shifts, this RNA ends up, tRNA without any amino acids, ends up in the E site for exit. This tRNA, which now has that polypeptide plus extra amino acid, ends up in the P site, and A site becomes free. Now, I know that it's really confusing. When we're going to finish in the lab, I will take 5 minutes of your time, less than that actually, 2 minutes of your time, and we will together demonstrate how translation works. Sounds good? I have a way to do that. So right now, if it's confusing, I understand. So how does it end? There's a special type of special molecule called a release factor. It enters the A site. It separates polypeptide from tRNA, and now you have polypeptide, and it's going to fold into a protein. This process, of course, requires energy. It's pretty fast. Okay? And as we mentioned, there are considerable differences between eukaryotes and prokaryotes. Okay? So in eukaryotes on the left, transcription and translation are separated. In prokaryotes, they are practically simultaneous. In eukaryotes, mRNA is processed. Splicing and other modifications. In prokaryotes, it isn't. Different ribosomes, and then eukaryotic mRNA is monocystronic, and prokaryotic is polycistronic. So let me explain what it means. So imagine you have 3 genes, 1, 2, and 3, in a eukaryotic organism, right? Does that make sense? In eukaryotic organism. If these genes are transcribed, they are transcribed into 3 separate mRNA. mRNA1, mRNA2, mRNA3. Each transcript is monocistronic. It carries 1 gene. In prokaryotes, however, same situation may result in a single mRNA. Where genes are not separated into individual molecules. So 1 mRNA contains 3 genes, or 4 or 5. Does that make sense? Now, proteins, when they're synthesized, can go through a variety of modifications. We aren't going to talk about these modifications because we have a chance to talk about herpes. Human herpes viruses are divided into 3 large groups: alpha, beta, and gamma herpes viruses. They are large. They have the envelope as the part of the virion, so they're not particularly stable. Okay? And they have enormous genome. Don't memorize any numbers. Please do not memorize the structure of the genome. Like, just follow what I'm telling you. The genome is double-stranded DNA. Okay? They have quite a few genes, up to 120. What is interesting about herpes virus infection, it takes its time. It's not fast. When I say it's not fast, like poliovirus, full replication cycle takes about 12 hours. Herpes viruses replicate a few days. They take time. Okay? Because they have so many genes, they have to time the expression of those genes along the infectious process. So there are immediate early genes that expressed first. These genes are required to, say, transcribe the viral DNA. There are early genes which now focus on DNA replication, and then there are late genes that complete the virus particle. Now, the process of infection is quite interesting. It's pretty common among viruses like this, but it's still interesting. So, first of all, any virus, and this is a law, any human virus that infects a human cell, or animal cell, or any animal cell, must bind to the receptor. These viruses have different receptors that they can bind to, and this determines which cells these viruses primarily infect in the body. So the virus binds to the receptor, the capsid gets into the cytoplasm where it releases double-stranded DNA. In the eukaryotic cell that you see on the picture, in a cell, in a human cell, where is DNA supposed to be? This particular DNA, like this one, is it in the nucleus? It's in the cytoplasm. It shouldn't be there. This is why there are mechanisms which move that DNA as fast as possible into the nucleus. Because if cell recognizes DNA in a cytoplasm, it's going to kill itself. It's going to go in apoptosis. So once the DNA is in the nucleus, it acts as the regular DNA. It's being, transcribed, translated, proteins are made, particles, capsids are made, particles are formed, and virus is released. Okay, makes sense? So basically, herpes virus DNA acts just like human DNA. The outcome is different, it's virus particles, but still. The unique feature, not unique, but distinctive feature of all herpes viruses is that they can become latent, dormant. So basically, herpes virus DNA stays in the nucleus in so-called episomal state. What does that mean? The linear DNA becomes circular and hangs out in the nucleus. And that's it. Nothing else happens. It just waits. A lot of textbooks, especially college textbooks, say that herpes virus DNA gets incorporated in the genome. This is complete and utter bullshit. It's not incorporated in the genome. It exists separately from our chromosomes, but it still sits in the nucleus. Right? So what does latency mean? If somebody acquires herpes virus, it goes latent, and there's a saying among virologists that what is the difference between the herpes and love? Herpes stays with you forever. Okay? So first let's talk about a couple of alpha herpes viruses that you know pretty well. Herpes simplex and varicella zoster, also known as chickenpox. So herpes simplex is known as labial herpes and genital herpes. Previously, HSV1 was considered the first type, herpes simplex type 1, was considered to cause cold sores, and HSV2 was considered to cause genital herpes. Now the idea, it's probably a mixed bag. Okay? Now, all herpes viruses can be transmitted with bodily fluids. We call it a kissing disease. All herpes viruses, well, most herpes viruses are transmitted in an early age among kids who have horrible hygienic habits. Okay? Can be transmitted through saliva, through urine, anything. And if you are a healthy person, the virus quickly becomes latent. Alpha herpes viruses become latent in neurons. Okay? When you become immunocompromised, let's say you had a cold, right? Your immunity is down. The virus reactivates. It reproduces and causes a cold sore. At that moment, if you have cold sores, this is when you are infectious. If you don't have, like I had cold sores since I remember myself. So I probably acquired it like a kindergarten, whatever. Maybe from my parents. Okay? So right now, I'm not infectious. If I would have a cold sore, I'm infectious. I can pass the virus to other people. Does that make sense to you? Okay. Other than that, we'll talk about how benign is that. Okay? But, okay. When we use the term immunocompromised, we usually refer to people who go through the cancer treatment or try, it's serious, right? If you I'm a fairly healthy dude. If I get a cold, I may get a cold sore after because my immune system was really busy. Okay? It will go away in a week. During that time, I'm infectious. Okay? I will not share my glass with someone. Okay? Something like that. But again, they're pretty unstable. So if you're concerned, oh, my partner has a cold sore, I probably should just absolutely fucking burn the house or burn the used disposable plates. We don't. Dishwasher will take care of it. Okay? Prevalence is pretty high. This shows you the percent of people who have HSV1 in China. Basically, by the age 60, everybody has herpes. In you're going to get it from other people. Immunocompromised folks, though, can develop pretty severe complications. Usually it's herpetic encephalitis, which is bad. The virus lives in neurons. It can travel to the brain, infect the brain. It's bad. Okay? It can be transmitted to kids through placenta. And neonatal herpes infection is awful, outright awful. Very severe, often leads to death. Practically every time leads to serious health complications in the future. Okay? Now, the disease that we don't see very often now is chickenpox, called by a varicella zoster virus. Okay? Produces typical rash. Okay? Some of you may have had it, some of you have been vaccinated. Vaccine is super efficient against the disease, and I can't emphasize it enough. Vaccines do not prevent infections. They prevent diseases. But, we're not kids anymore, so at my age, I should be concerned eventually about shingles, which is an example of the reactivation of chickenpox. Go ahead.
I know that sometimes younger people can get shingles.
They can. They can, absolutely. It is rare in younger folks, but it can happen. Absolutely, 100%. But shingles vaccine is recommended for everybody over the age of 55. I urge you to, nobody here is over the age of 55, so I urge you to take it when you get there. Okay, I'm definitely going to take it because a colleague of mine didn't. And he had shingles in his eyes. Yeah, it can infect the cornea, yes. Got corneal infection and lost some of his vision. So it's bad, yeah. And this hurts a lot. Also, I never had a vaccine, and I never had the manifested chickenpox, which means I was infected. I never got sick because my wife had, as an adult, she had chickenpox as an adult. And I was as close as you can be to your wife. I definitely was infected. I did not have any symptoms of infection. Okay? One interesting thing about herpes, and I told you it's going to be interesting. There are a lot of people who are infected with herpes, a lot of people who have had chickenpox, or people who had shingles, right? There are several studies, and by several I mean more than 10, that quite convincingly established the link between herpes and Alzheimer's disease, or herpes and or chickenpox and general dementia. One of the most recent papers, it's a very coolest study, just coolest study. It was done on older people. There was a population of people who were not eligible for varicella zoster vaccine, okay? Say, if you're 65, you're not eligible. But if you're 64, you are. So they 65 and 64, that's pretty close age-wise, right? So they compared the rates of dementia in people who received and who didn't receive varicella zoster shingles vaccine. Those who received it had lower rates of dementia. Yeah, there's a plenty of evidence towards that. And another study looked at progression of Alzheimer's in two groups of people. One was just fine, just leave. And another group was treated with acyclovir. I think it was acyclovir. No, it was some other drug, but basically acyclovir is injections, and it was a version that is oral. So they were given oral antiherpetic drug. People on that drug showed slower progression to dementia. So, interesting. Now, beta herpes virus that is worth our attention is cytomegalovirus. So, I worked in it for 5 years. In immunocompetent adults, absolutely benign. Does nothing. It's still debatable if it is associated with cardiovascular risks. For 5 years, I was trying to show that it is associated. Weak. at best. We did it in animals, so not even in humans, okay? Yeah, so you get it just like any other herpes virus, bodily fluids, and nothing happens then. Maybe a little bit of a cold. If you become immunocompromised, though, super bad. Infects pretty much everything from, it's normally latent in monocytes and blood cells, but if you're immunocompromised, it gets out of the monocytes and infects all kinds of cells, and it's very common in patients who have AIDS. But it is famous, well, infamous, for neonatal infections. So this is a clinical case. This girl was born with congenital cytomegalovirus. She died 6 days after birth. And honestly, I think it's the best outcome. Let me explain. Because congenital CMV look at cognitive deficits, hearing loss, microcephaly. That's the percentage in children. It is like, it's horrible. It's very bad. Okay? And unfortunately, the current administration, the current head of Health and Human Services, Department of Health and Human Services, ordered to cancel funding for all mRNA vaccine studies. One of the mRNA vaccines that was actively in development in clinical trials, the vaccine against CMV, that would be given to women early in pregnancy to protect them from CMV, because if a pregnant woman gets infected with cytomegalovirus, she's fine. But it will cross placenta and infect the fetus. So I don't know if they get to the phase 3 and if they will ever manufacture, because that would be an incredible benefit for, reducing number of babies born with lifelong disabilities. Okay? Now, maybe a little less known, gamma herpes viruses, which are Epstein-Barr virus and Kaposi sarcoma herpes virus. So let's talk about Epstein-Barr. Are you familiar with the disease known as mono, infectious mononucleosis? It is caused by the Epstein-Barr virus. So Epstein-Barr virus, when it infects humans, it infects cells in the pharyngeal epithelium. So this is why you have the sore throat, that kind of respiratory symptoms, right? And it also infects immune cells, B cells. So this is why people have swollen lymph nodes, swollen tonsils, and then it goes away. But virus EBV remains latent in B cells. And it's been very clearly shown that Epstein-Barr virus, latent Epstein-Barr virus, later in life can cause Burkitt's lymphoma. It's a B-cell lymphoma. And remember, it stays latent, like close to your nasopharynx. It can cause nasopharyngeal carcinoma. In the last 15 to 20 years, it's been associated with a bunch of cancers, like they generally called head and neck cancers, like throat cancer, oral cancer. And it looks like Epstein-Barr virus teams up with human papillomavirus in inducing. Like, one virus is not like super risky, but when you have two, tough luck. Okay? Now, I'm going to say a few words about Kaposi sarcoma very briefly and then quickly go back to EBV. So Kaposi sarcoma, this blotches in the skin, it's sarcoma, it's a connective tissue tumors. It is cancer caused by a virus. It happens almost exclusively in immunocompromised people, AIDS patients. It is known for being absolutely notorious in AIDS patients. Why I wanted to go back to Epstein-Barr. Mono, right? So last year, a study was published. And in that study, they looked at a bunch of people who were in the VA system. They looked at health records of those people. Because if you serve, then you can receive your medical services in the VA system as a veteran, right? So the health records can span tens of years. They screened, I think, 2 million records. Like, it's a pretty sizable cohort. And they found a very strong association between having mono and developing multiple sclerosis later. People who had mono had higher chances of developing multiple sclerosis. Don't ask me about mechanism. Nobody knows. But it just, probably some immune dysregulation. But it just tells you that when we think about like a virus infection, like we think, oh, it's mono, okay, I'm fine. Maybe not. I have a cold sore. Maybe I will develop dementia in 9 years. Who knows? Okay? So just the consequences are really interesting. All right, folks. We're going to move on to upstairs.